in clinical psychology and also her PhD at the University of New South Wales in Sydney, Australia. And after working in the field of developmental psychology, she completed a postdoctoral fellowship at Columbia University with, I believe, Nim Tottenham. And so I was quite pleased that since we had lost Nim Tottenham to Columbia, that we were able to recruit her here at UCLA. She joined us in 2019. She's the director of the Brain and Body Lab. And as you will hear her talking about, her work is really addressing how early caregiving environments influence the development of mental and physical health, and how interaction between the brain and body contribute to health and disease outcome. Now, she is being funded by NIMH, as well as the Brain Behavior Research Foundation and the National Health and Medical Research Council. She has received an Early Career Award from the Federation of Association for Brain and Behavioral Sciences. I'm sure I'm not going to pronounce it right, a Katersky Young Investigator Award from the International Society for Developmental Psychobiology. And she was also named a Rising Star by the Association for Psychological Sciences, APS. So without further ado, it is please join me in welcoming Bridget. We're all looking forward to hearing your work. Thank you. Yeah.
Did I just turn that off? Oh gosh. Can you still hear me online?
Yes.
Yes.
Great, thank you. Appreciate that. Okay, so thank you for that lovely introduction, and also thanks for the invite here. I'm really excited to share with you some of my work on the intergenerational impacts of adversity on mind and body health. I have a thing, so I can use this. Okay, so my research program is really centrally focused on this relationship, the attachment relationship between a parent and a child, a relationship that, when it is secure and strong, can really help children deal with all manner of challenges in this world. And when it is disrupted or ruptured, can really predispose individuals to exhibit difficulties across really basically all areas of their life. So we call adversities that happen within this relationship caregiving related adversities. And they can constitute things such as parental abuse, neglect, separation from the caregiver. So we know that disruptions to this caregiving relationship, either in the prenatal or in the postnatal period, what's happening? Okay, now we work. Okay, right, good. Prenatal or in the postnatal period are associated with an increased risk of nearly all examined adult mental illnesses. So they account for up to 30% of adult mental illnesses. And they're also associated with an increased risk of a ton of physical health problems, including things as diverse as heart disease, respiratory disease, and also gastrointestinal problems. Now, critically, the impact of adversity does not stay with the individual who is directly exposed. In fact, there's a lot of evidence to suggest that there's a perpetuation of adversities which occurs across generations. This has been most famously shown at the population level, with children of individuals who experience things like starvation or war or survivors of genocide also being at increased risk for physical and mental health problems themselves, despite having never been directly impacted. But we also see evidence of this with things like caregiving adversity. So given this information, it really highlights for me that there are two major priorities that we should have right now in our understanding of well-being. And the first is that we, of course, need to be thinking transdiagnostically. So what are the mechanisms which can explain the diversity of symptom profiles which are commonly observed after adversity exposure? And that includes both within mental health, so for example, the coincidence of things like anxiety and depression, as well as between mental and physical health. So for example, the coincidence of things like anxiety and gastrointestinal disorders that are functional in nature, like irritable bowel syndrome. And then the second priority, it suggests to me, is that we really need to understand mechanisms which could potentially account for the transmission of risk across generations after adversity exposure. And I think that having both of these foci is really going to help us get to a place where we can better address the multitude of adverse health outcomes that are attributable to adversity exposure. So in terms of these transdiagnostic mechanisms, I have a particularly strong interest and a growing interest across my career in this axis here, the brain, gut, and microbiome axis. Now, this is a bidirectional communication highway. Communication occurs through a multitude of pathways that I'm not going to go into, but they do include vagal and spinal afferents, as well as a ton of chemical signaling that's going on. And I'm interested in this pathway because it is transdiagnostically associated with a range of different mental health as well as physical health problems. And also critically for this talk, there is some emerging evidence suggesting that this brain-gut microbiome axis is affected by early life caregiving adversity. So what I'm going to show you in the talk today is data stretching across 2 different generations of humans. And it's going to be a talk in 4 parts. So in the first part of this talk, I'm going to ask how early caregiving adversity experienced in this first generation affects brain development and how that brain development is associated with youth mental health problems. In part 2, I'm going to move away from that kind of exclusive focus on the brain and begin to ask questions about how caregiving adversity affects this wider brain-gut microbiome axis in ways that might be important for mental health. In part 3 of the talk, I want to ask how adversity experienced in generation 1 can affect the brain as well as the wider brain-gut microbiome axis in the next generation, in generation 2. And then finally, in the 4th part of the talk, if we have time, I'm going to tell you about some more recent work we're doing, looking at how caregiving adversity affects the way that one transitions into motherhood and the risk for perinatal depression, which we know is a significant factor involved in the perpetuation of adversity across generations. So without further ado, I'm going to dive into the first part of the data here, looking at the impacts of caregiving adversity directly experienced on brain function. So what we're going to focus on here is the impacts of adversity on amygdala development. So you might ask why amygdala development, although I'm sure most of you are probably on board here, but numerous studies have shown that caregiving adversity really does have an outsized impact on limbic system development, including the amygdala. And this is likely because these regions undergo really rapid structural growth during early life when children are most dependent on their parents, on their close others, for their emotion regulation. So if these brain regions are going to be responsive to any environmental cues or signals to guide their development, then caregiving adversity is very likely to be one such cue. Indeed, studies have shown that the amygdala is particularly responsive to caregiver cues in the early environment. And this was an effect shown by Dylan G, published in 2014 in Psych Science. So what they did in this study was have children lie in a scanner and look at pictures of either their parent's face or a stranger's face. And then they simply contrasted amygdala reactivity to these 2 different faces, so parent minus stranger. And what they found was that specifically in childhood, so the black bar, but not in adolescence, the white bar, there was lower amygdala reactivity to pictures of parents relative to strangers. And they interpreted this as amygdala buffering to parent cues, so downregulation of the amygdala to these parent cues. So this really suggests that the amygdala is responsive to caregiving cues in the environment in childhood, but as that child turns into an adolescent, as they increase their independence from the caregiver, then the amygdala becomes somewhat less responsive to those parental cues specifically. So if that's true, then it would suggest that a lack of caregiving cues in the environment from early in life, such as when you're separated from a caregiver, might affect what the amygdala begins to respond to. And we suggested that it might actually decrease the amygdala's reactivity to parent cues even earlier in development. So during my postdoctoral training, which I did in Nim Tottenham's lab, as Marilla noted, I had access to some of these data. And so I wanted to ask that question. So how does exposure to caregiving adversity affect amygdala responsivity to parent cues in childhood? So I had that exact same task, but this time we had a group of children who were previously institutionalized, or PI, which means they were raised in institutions or orphanages abroad before being adopted into American families. And for these youth, they were looking at pictures of their adoptive parent or a stranger in the scanner. And what we found is that these youth did not, at the group level at least, show amygdala buffering to those adoptive parent cues. And this was not associated with age at the time of adoption or the amount of time spent with these adoptive caregivers. So this suggests that caregiving adversity does disrupt the responsiveness of the amygdala to parent cues in childhood. And individual differences here, and you can see based on the size of these error bars within the PI group, they were significant. They were also clinically relevant. So specifically, what we're looking at here is anxiety on the y-axis, and then we've got the comparison and the early life adversity groups on the left and right panels. You can see that in general, regardless of whether individuals showed amygdala buffering to parent cues at time 1 within the comparison group, they showed low anxiety both at year 1, which was the time of the scan, as well as at year 3, so 2 years later. Whereas for the early adversity group, their amygdala buffering at time 1 really seemed to be associated with what their anxiety did over time. So those individuals who were able to buck the trend and show amygdala buffering to parent cues showed a sharper decline in anxiety across time. And this was also related to attachment security. So those previously institutionalized youth who reported more security in their attachment relationship with their adoptive parent were also in the buffering at time 1 group. So they also showed that amygdala buffering. So this suggests that individual differences in amygdala buffering to parent cues are relevant for affective functioning across development, and importantly, that they could potentially be targeted through parenting-related interventions that increase child's felt security in the attachment relationship. Now, in another study, we've shown that while parent cues no longer buffer or decrease the reactivity of the amygdala in adolescence, parent-adolescent interactions still do shape amygdala reactivity in ways that are associated with adolescents' mental health. So in this 7-year longitudinal study that I did with Dr. Sarah Whittle's group at the University of Melbourne in Australia, we looked how aggressive parent-adolescent interactions when youth were 12 years of age were associated with amygdala connectivity patterns, resting state, when they were 16 years of age, so mid-adolescence, and then whether that mediated the association between aggressive mother-adolescent interactions at 12 years and late onset depression diagnoses when adolescents were at 19 years of age. And so what we found in this study was that maternal aggression was related to resting state functional connectivity between the amygdala and the right superior temporal posterior insula, the bilateral visual cortex, and left temporal and insular cortices, and that this left temporal and insular cortex was actually associated more strongly with the centromedial subdivision of the amygdala. And critically, it was this region circled in red here that mediated this association between maternal aggression and late adolescent onset diagnoses of depression. So again, parent-child interactions from early in life through adolescence shape amygdala circuits in ways that seem to affect later emotion regulation and mental health. And there are some clear ideas here for intervention because positive parenting in both instances might have a real impact on children's amygdala responses in ways that could potentially impact youth mental health. So far we've looked at the impact of, or the association of, experiences of early life adversity and brain function. But one underexplored possibility out there in the world is that early life caregiving adversities may increase the risk for youth psychopathology through a widespread impact, not just on the brain, but also on peripheral systems that are involved with health. And one of the peripheral systems that is of particular interest to our research group is the microbiome and the wider brain-gut microbiome axis. So that's what we're going to look at here. So as I mentioned at the start of this talk, several studies have already shown that early life adversity is associated with changes in the microbiome. But most of those studies, including my own, have been small sample sizes, or in the larger studies, they've been adults retrospectively reporting on their own experiences with childhood adversity. And all of the literature so far has focused on the gut microbiome, neglecting other microbial communities that may play an important role in health. So in this next section, I want to show you what we're discovering about how adversity affects different stages of how adversity at different stages of development affects the microbiome, and then how that links to youth mental health outcomes. So the first metric that I want to look at here is called alpha diversity. This is a within individual metric looking at how rich and evenly distributed an individual's microbes are in their own gut, which we then obviously average across groups. Now, typically, alpha diversity is very low early in development, and there is an enormous increase in alpha diversity across the first 3 years of life, where it seems to then stabilize in adult levels. So what we see in the context of postnatal adversity exposure is that this trajectory might be accelerated. So specifically here, we had postnatal adversity we were defining as high levels of maternal anxiety and depression, so actually clinical levels. And this was associated with a sharper increase in alpha diversity from birth to 2 years of age. And so for this data set, we have 100 individuals with no missing data across 4 time points in the first year of life. Moving beyond 2 years of age, we see that postnatal adversity exposure seems to change the trajectories of alpha diversity even into adolescence. So here, the caregiving adversity was a little different. So here in this study, caregiving adversity exposed youth had been maltreated and then were adopted from foster care. In the comparison group, you can see that there's relative stability in alpha diversity from 2 to 17 years of age, which was as expected. However, in the adversity exposed group, we found something really quite unusual, which is a non-linear decrease in alpha diversity with age, whereby the lowest levels were seen in the adolescents. This is a very, very strange trajectory. It hasn't been looked at before. Really, no one is looking at moderators of age-related change in the microbiome as far as alpha diversity goes. So this was pretty interesting to see. Now, in this same study, we saw nonlinear changes, again, in the microbiome when we look at the single taxa level. So instead of looking at something like alpha diversity, which is the whole community, if you look at the relative abundance of single microbes. So we actually saw 12 microbes that showed age-related change in relative abundance that was moderated by adversity exposure. But because that doesn't fit on the screen, I'm just showing you 3 here as examples of this effect. So here we've got age on the x-axis and microbes relative abundance on the y-axis. And we've got the comparison group in blue and then the adversity exposed kids in orange. So the first thing I want you to note here is in the blue. So the microbiome is changing a lot across the adolescent or across the early childhood through adolescent stages of development. Sometimes that change is linear and sometimes it's a non-linear change. And I mention this because there's actually a pretty strong narrative derived from those studies in alpha diversity showing that the microbiome is done developing by 3 years of age. And clearly that's not true of alpha diversity when we look in groups of individuals who differ from the norm, so in adversity exposed youth. And it's also not true of the relative abundance of single microbes, either in the comparison group or clearly in the adversity group. Because of course, the next thing to note here is that adversity changes these trajectories of single microbe abundance, and it does so in different ways. So now comes the next kind of obvious question, which is why should I care? Or why should you care? Now, of course, the microbiome is interesting for microbiologists for a variety of reasons. But when it comes to psychologists and psychiatrists, we care about the microbiome because of its links to mental health. So we want to see whether any of these microbes are linked to mental health. So within the 12 that showed a significant age-related change that was moderated by adversity exposure, we looked at all of them to see what their associations with internalizing symptoms were, so anxiety and depression symptoms in kids, and we corrected for multiple comparisons. And the reason that I'm showing you these 3 here are because these are the 3 of the 12 that had significant associations with internalizing symptoms in youth. So specifically, we found here that the first one, which is called Clostridium inocuum, which tended to be higher on average in the adversity exposed group, was positively associated with internalizing symptoms. And this was a pretty sensical association for us because Clostridium inocuum is typically considered to be a pathogenic species in the gut. Eubacterium hallei, which again tended to be slightly higher in the adversity exposed than the comparison group, was also positively associated with internalizing symptoms. Though this was a little bit harder to interpret because groups of hallei seem to be butyrate producers, and we tend to think of butyrate as being fairly positive, both in the gut as well as in the brain and wider body. And then the final microbe that was associated with internalizing symptoms is Butyrosemonas, which tended to be lower on average in the adversity exposed group and was negatively associated with internalizing symptoms. So we were really interested in this one because it seemed to be potentially a protective microbe. So the final question comes then, which is now that we see that these microbes seem to be associated with age, they seem to be associated with adversity, and they're associated with health outcomes that we care about, what can we do about it? Now, while many people in this space are very interested in probiotics, and I do think the probiotics field is quite interesting, In fact, there are simpler interventions. Diet, for example, is a non-invasive and inexpensive way of altering the microbiome that is, fairly tolerable for young children. So before we go in and do a diet intervention, we just wanted to look and see whether any of these associations here were moderated by diet. So these data were analysed by fantastic graduate student Nomi Gantz in the lab. And interestingly, Nomi found that fibre specifically was associated with the enrichment of Butyromonas, that potentially protective microbe that we saw earlier, being negatively associated with internalising symptoms. And it was negatively associated with Butyromonas specifically within the, sorry, I'm saying that the wrong way. It was positively associated with, fibre was positively associated with Butyromonas specifically within the interpersonal adversity exposed youth. And to kind of follow up on that finding, supporting that association, Nomi found that fibre consumption itself was negatively associated with internalising symptoms. And again, this was specific to the caregiving adversity exposed group. So this suggests that fibre has unique beneficial relationships with mental health related microbiota within adversity exposed youth, highlighting the potential for future nutritional interventions that may be especially salutary for adversity exposed youth. So while the gut microbiome community is absolutely the darling of the microbiome world, there are other microbial communities that exist outside of the gut and that have been linked to mental health and which might be affected by caregiving adversity. So it's important for us to look and see what those are. So what do we know about the next largest microbiome community in the body after the gut, which is the oral microbiome? Well, there has been some attention to this community, and there's one study published in 2021, which has shown that the oral microbiome of adults who had experienced early life caregiving adversity was disrupted. In this particular study, they didn't see any differences at the community level, so no alpha and beta diversity differences, but they did see a lot of differentially abundant microbes that were associated with prior exposure to caregiving adversity. So Nomi, again, my graduate student, was really interested in looking at the impacts of caregiving adversity earlier in development, so when children are still young. And she wanted to zoom in on pathways that intersected with the stress hormone cortisol, as we know that this is dysregulated in the context of early life adversity. And there should be associations between cortisol and microbes across the body, including in the mouth. And Nomi had this really interesting hypothesis that the oral microbiome of adversity exposed youth might become insensitive to cortisol, as previous studies have shown the same with immune cells. So immune cells do become less sensitive to the regulatory effects of cortisol in the context of caregiving adversity or CA exposure. So in this particular study, Nomi had access to hair cortisol, which indexes tonic exposure to cortisol over the last 3 months, so more of a recent cortisol exposure. And then she used this hair cortisol as a moderator in models examining the association between early life adversity and the oral microbiome. Now, remarkably, Nomi found essentially the same thing as had been shown with immune cells within the oral microbiome community. So specifically, she saw that whereas the oral microbiome community of comparison youth did seem to be regulated by hair cortisol, so higher hair cortisol was associated with lower alpha diversity, there was no association within the caregiving adversity exposed youth in orange between hair cortisol and alpha diversity. Now, outside of the influence of cortisol, Nomi also found that there were several taxa which were associated with caregiving adversity, including this Porphyromonas species, which is a pathogen in the mouth, which was higher in the adversity than in the comparison group. And critically, this Porphyromonas species was positively associated with children's internalizing symptoms. So again, this suggests that even microbial communities outside of the gut, which are affected by caregiving adversity, these may also play some role in youth mental health. And the exciting thing about this is that it really opens up the door for even more pathways to potentially influence the microbiome and thus indirectly chip away at youth mental health or increase youth mental health. And even simpler than dietary changes, the oral microbiome community can be affected by things as simple as oral health behaviours like toothbrushing. So now I've shown you that directly experienced caregiving adversity in childhood impacts the development of children's gut microbiome and community composition and the differential abundance of microbes in the gut. And it also affects the way that the oral microbiome responds to cortisol and the differential abundance of microbes in the mouth. And I've also shown you that some of these microbiome outcomes are associated with youth's mental health. But one major issue remains, and that is how. So how would the microbiome, these bacteria in our guts and in our mouths, influence something as complex as mental health problems? Now, of course, there are a multitude of ways that microbes in the gut could be linked with youth mental health, and all of them, of course, involve the brain. So now we have a problem where we are trying to link to incredibly rich and complex, multivariate, highly dimensional organs with one another, the brain and gut microbiome. And we want to do this in a way that tells us something meaningful about a heterogeneous, non-distinct clinical symptom cluster like internalizing symptoms in youth. And we want to do this all non-invasively in small humans who don't like to lay still in an MRI scanner. So, fun times. So how are we ever going to make progress on that front? It's going to be a tough one. And I want to show you one of our first true attempts at doing this using a top-down, multivariate and longitudinal approach. So in this particular study, we had 55 complete cases with the following measures. We had microbiome when youth were 2 years of age. We had high-quality inter and intra-resting state network connectivity when youth was 6 years of age. And then we had internalizing symptoms, so anxiety and depression when youth were 7.5 years old. So here in this multivariate and data-driven, symptom-informed longitudinal approach, we had 3 steps. In the first step, we used something called multivariate sparse partial least squares regression, or an SPLS, to determine a linear combination of brain networks or a component that best explained variance in internalizing symptoms. We then used just those component scores as the outcome in step 2. And now we're asking whether what linear combination of microbes best predicts variance in these internalizing associated brain networks. And then in the final step, we took the outputs from step 1 and step 2 and fed them into a mediation, where we're asking whether the microbiome patterns identified in step 2 were indirectly associated with youth internalizing symptoms via the brain patterns that were identified in step 1. For the sake of time, I'm only going to show you the most interesting data here, though it's worth noting that we did see a range of different brain networks, a range of different microbe networks, but really only one that seemed to work out in the mediation, which is the one I'm going to show you. All right, so in step 1, what we found, one of the things we found was this linear combination of resting state brain networks, which we called the SOFA signature, which explained maximal variance in internalizing symptoms in youth. And this was characterized by connectivity between the SOFA network, which is the striatal orbitofrontal amygdala network, and a variety of other brain networks, including the frontoparietal network and the default mode network. So as you can probably tell, for those of you who do brain research, which I presume is everyone, the striatal orbitofrontal network, very important for, mental health and emotional health in general. So it was not a particularly surprising pattern to come up here as seeing a lot of SOFA connectivity. So then in step 2, what we did was we used SOFA connectivity as the thing we were predicting, and we asked what linear combination of microbes best predicted variance in those networks. And what we found was this really interesting pattern show up, which was characterized by high positive loadings from Veillonella and Intestinibacter. And based on the prior literature, these 2 microbes have been associated with intestinal inflammation and have also shown to be higher in depressed adults. And it was also associated with high negative loadings from Fusocatinibacter and Catinibacterium, which have also been associated with depression in adult samples. So then in the final step here, we tested that mediation, and we did see a significant indirect association between the microbiome component identified in step 2 with internalizing problems identified at 7.5 years of age via the SOFA network connectivity patterns when youth were 6 years of age. And critically, this mediation is specific to this pattern. So although we defined the microbes based on the based on the brain and the brain based on the internalizing symptoms. We did that for everything, and only this one shows up as significant in the mediation model. Okay, so as a final summary of part 2, I've shown you that directly experienced childhood caregiving adversity impacts that gut and oral microbiome development, some of which seem to be associated with youth mental health outcomes. And now in this most recent study, I've shown you that we can work backwards from mental health to identify relevant brain networks and then microbes that are associated with those brain networks. And this is important because it might give us something to target in therapy. So if we're able to work backwards in this way, we may be more readily able to get at microbes that could be potential targets for therapy. All right, so in part 3 now, I'm going to switch gears a little and talk across generations. So looking at how caregiving adversity impacts the next generation. So focusing first on the brain and behavior of these second generation youth. In this study, I wanted to look at the impacts of maternal experiences of childhood adversity on second generation children's brain and behavior. So these data were analyzed by Dr. Jessica Wee, who is now at Stanford University. She was a postdoc in the lab at the time. And we had access to a data set where we had 541 mother-child dyads with the following data points. We had mums who had retrospectively reported on their own experiences with childhood trauma, which tended to be mostly emotional and physical neglect, and this was present in about 20% of the population. We then had mums' maternal prenatal mental health and mums' postnatal mental health symptoms, which could potentially be considered as adverse experiences for the fetus and for the young child. And then we had children's emotional health outcomes when they were 7 years of age, which were both parent proxy reported and self-reported by the child. So what Jess found in this study was that maternal childhood trauma was related to worse parent proxy reported child emotional health outcomes when youth was 7 years of age, and that this occurred serially through a pathway of worsened maternal prenatal and postnatal mental health problems. She also saw that maternal childhood trauma in the mother was related to child self-reported emotional health, and this occurred through a pathway of worsened maternal prenatal mental health outcomes. In a subset of these data where we had high quality resting state functional connectivity when youth was 6 years of age, Dr. Wee then looked to see whether there were any relationships between any of these different experiences and children's amygdala prefrontal cortex resting state connectivity. And she focused in on amygdala prefrontal cortex networks because of their extensive relation to children's emotional health and the development of self-regulation. And what Jess found was that over and above maternal childhood maltreatment and maternal prenatal mental health, maternal postnatal mental health had unique associations with children's amygdala PFC connectivity. Specifically, higher maternal postnatal mental health was associated with lower or more negative amygdala PFC functional connectivity in those second generation children when they were 6 years of age. And this functional connectivity pattern in turn was associated with both parent proxy reported and child self-reported emotional health problems when youth was 17 years of age. So this suggests that there are intergenerational impacts of adversity on children's emotional health and on their brain function. For the behavioral effects, so when we're looking just at emotional health outcomes, these seem to be mediated through maternal mental health in both the prenatal and the postnatal period. But when we're looking at children's brain function, this seems to be more closely tied to the most proximal relationship to the child, so maternal postnatal mental health since the child's birth. So in thinking of this idea of transdiagnostic mechanisms, my graduate student, Fran, was really interested in using this same cohort to understand whether there might be an impact of childhood adversity in the mum, prenatal and postnatal maternal mental health on children's microbiome in the second generation. So here she had 450 dyads from this same cohort who had gastrointestinal microbiome measures at 2 years of age. And in this study, as in all of our microbiome studies, she controlled for important things that impact the microbiome, like diet, ethnicity, sex, duration of breastfeeding, etc. So what did she see in this study? There were quite a few different things. So the first thing Fran looked at is something, again, called alpha diversity. So remember, this is the community metric, a within individual measure of the richness and evenly distributed microbiome. And she found that both prenatal adversity and postnatal adversity, but not maternal childhood adversity, were associated with children's alpha diversity at 2 years of age. So specifically here, we see two things. We see that higher maternal prenatal adversity was positively associated with the evenness in the distribution of the microbiota at youth 2 years of age. This suggests that potentially exposure to prenatal adversity may accelerate the development of alpha diversity in the gut. And then she found that for postnatal adversity, having high levels of postnatal adversity was negatively associated with another measure of alpha diversity in the gut. So again, potentially hinting at these dynamic changes in development of the gut microbiome following adversity exposure. There were no impacts of any of these forms of adversity on beta diversity, which I'm not going to get into, but is a between individual measure. However, when we looked at the individual the differential abundance of individual microbes, single taxa, we found that each of these forms of adversity were associated with a range of differentially abundant microbes. So here I have those microbes plotted. Those on the right of the line are more abundant with more adversity exposure, and those on the left more abundant with less adversity exposure. And I've color-coded them according to the different types of exposure. So preconception is mum's childhood adversity, prenatal during pregnancy, and then postnatal after birth. So what we found in terms of the prenatal adversity first, we found two types of streptococcus were higher with more prenatal adversity exposure, and these are part of the normal microbiome, but several species are pathogenic, so that was interesting. We then saw ruminococcus lower in the context of high prenatal adversity, and several of these species are associated with immunity, both anti and pro-inflammatory effects, as well as with dopamine. So this is a little harder to interpret. Parabacteroides rather, was lower in the context of high postnatal adversity. This is generally considered to be a probiotic, or many species are considered to be probiotic. So that made sense here to be lower in the context of high postnatal adversity. And high postnatal adversity was also associated with higher Finagoldia, which is a normal part of the microbiome, but an opportunistic pathogen. And then finally, and for us, the most interesting finding was that Clostridium sensu stricto was higher in the context of preconception adversity, so mums who had high levels of childhood adversity exposure. Now, this is an interesting microbe because it is a butyrate producer, but it actually is a rather inefficient butyrate producer, and it really should have been outcompeted by more efficient butyrate producers by around 2 years of age, based on the literature. So we think that this may hint at maybe slightly more inefficient butyrate production in youth whose mums experienced childhood adversity. So based on these data and in the context of our earlier findings, we're again seeing butyrate production cropping up as something that seems to be associated with early life adversity, at least in terms of the functions of the gut microbiome. And we're also seeing hints here that immune-related functions may be disturbed in the context of generational adversity. Now, it's really hard to tell here, as we have to have a guess at functions, because the type of sequencing that we did with these data, with these stool samples, was 16S, which only gets us down to about the genus level. So there's not a huge amount of specificity that we can derive from these data. However, we do now have metagenomic sequences on all of these samples and actually all of the data that I've shown you. So we'll now be able to directly or more directly address the potential functions that may be preserved in the gut or may be altered by adversity. So we can stay tuned for that. So the summary of part 3 is that maternal childhood adversity seems to affect children's behavior and brain via impacts on maternal prenatal and postnatal mental health problems. And it also seems to affect children's microbiome via pathways that partially overlap with those of prenatal and postnatal maternal mental health. So given these findings and the clear importance of maternal mental health in intergenerational impacts of adversity, we have become increasingly interested in this person here, the mother, specifically while they're pregnant, as a conduit for these intergenerational effects, and specifically thinking about how mum transitions into motherhood and the impact of this on her mental health as being a really critical player here for these generational impacts. And so for the last very few minutes, I'll probably only take 5 minutes for this, so we'll still have time for questions at the end. I'm going to zoom through some very recent data we've got looking at how caregiving adversity impacts the transition into parenthood. So zooming through this, pregnancy is really a critical period of development, which is characterized by heightened risk for depression. And women who are exposed to childhood trauma are at greater risk for peripartum depression. And this is important not just for the mums, which is reason enough, but it also affects the children. So peripartum depression could perpetuate cycles of adversity, leading to these intergenerational impacts. Now, it's been convincingly argued that depression at other stages of the lifespan arises from disordered interoception, which is the way that the brain perceives, integrates, and models sensory signals that are generated from the body. And while some differences exist between depression in the perinatal period and at other stages of life, both are really characterized by that core negative affective symptoms, low or irritable mood, as well as abnormalities or altered functioning across numerous bodily systems. And it's this abnormal functioning across the brain and across the body that suggests that depression is really a disorder of inefficient energy regulation. Now, energy regulation is supported by interoception, and thus it won't come as a surprise to you that several studies have now shown that interoception is impaired in the context of depression. So in this last set of analyses, we would have begun to wonder whether interoception might also underlie depression during the perinatal period. So when we think about pregnancy, one thing is for certain, and that is that interoception absolutely should change. Interoception, as a reminder, is that process by which the brain models sensory information from the body. And there's no other stage of the lifespan, maybe apart from very early infancy, in which sensory information generated from the body is undergoing such rapid and massive changes. This is a very compressed period of drastic brain and bodily changes. So this is a conceptual figure from a theoretical review that we just had published in Neuroscience and Biobehavioral Reviews, where my fabulous graduate student, Paul Savoca, is leading this work. And here we've suggested that under normal circumstances, interoception should increase across pregnancy to keep up with these high metabolic demands, which begin to show in pregnancy and decreased slightly postpartum. And interestingly, studies have shown that early life adversity exposure is actually outside of pregnancy associated with lower levels of interoception. So our theoretical model suggested that these may become amplified during pregnancy to lead to a higher risk for peripartum depression, specifically in early adversity exposed women, which would be mediated through impaired interoception. We've since found support for these hypotheses. So this was published just very recently in the Archives of Women's Mental Health, again, led by Paul. And basically what he's finding in this study is early life adversity moderated differences between pregnancy and comparison groups on 3 different measures of interoception. So from this one scale of interoception. So attention regulation, which is the ability to sustain attention and control bodily sensations. Self-regulation, which is the ability to regulate distress by attending to your bodily signals. So if you think, I need to breathe in to calm down. And then noticing, which is the awareness of body sensations, regardless of valence. And so what Paul finds in this study is that for people with low levels of adversity, so you can see highlighted in yellow the regions of significance here. So for people with low levels of adversity, we actually seem to see a boost in interoception for the self-regulation and the noticing subscales of the Maya within the pregnancy exposed group. So this, along with our hypotheses, suggests that pregnancy may be a period of time under normal circumstances where we can boost interoception to keep up with metabolic demands. However, he also found that for people who had high levels, mothers who had high levels of early life adversity exposure, they had lower levels, the pregnant women had lower levels of attention regulation and noticing. Again, suggesting that early life adversity seems to impair interoception, specifically in pregnancy. And critically, we found that both of these subscales, so the attention regulation and the noticing, were associated with depression. So more, the self-regulation and attention. So more self-regulation, more attention is associated with lower levels of depression. So this suggests that interoception might impart buffering effects against depression, which are boosted during pregnancy for individuals with low adversity exposure and weakened for individuals with high adversity exposure, which may be a pathway to elevated perinatal depression, specifically in adversity exposed women. So to summarize this last section, maternal childhood adversity affected mothers' experience of interoception changes during pregnancy in ways that are important for maternal mental health. And it suggests to us that focusing on interoception during pregnancy might be an important way to address these intergenerational patterns of trauma. And that's critical because interoception has been shown to be fairly easily targeted through a range of non-invasive treatments, including certain mindfulness-based therapies, as well as interoception targeted training. So returning to our research priorities of transdiagnostic mechanisms and the importance of intergenerational experiences, we think that focusing on features of an individual's biology and psychology that integrate the brain and body, so specifically looking at the brain and the microbiome and the brain-gut axis, as well as interoception, and focusing on mum's mental health during this pregnancy period where there's high metabolic strain is going to go a long way in helping us to improve individual well-being after adversity exposure. So I wanted to acknowledge the fabulous students who've done all of this work. So Fran and Nomi and Genesis are our microbiome mavens. And then we've got Dr. Jess Wee, who is our brain imaging buff, and then Paul Savoca, who is our wonderful interoception guru. Those are not the only students we have in our lab. We did manage to get everyone together and finally got a good picture. So here it is to our collaborators and our funders. And I also wanted to show you my own little dude. I managed to get a scan of an MRI scan for a study when I was pregnant with Freddie. And I love this one because you can see both of his brain and his gut in the same picture. And this is him much later in life at three years old. So thank you very much and happy to take any questions anyone has. Yeah.
Thank you. That was an amazing talk. My question was, I know you talked a lot about generational impacts of like maternal childhood trauma on the child, but I was wondering if the microbiome starts changing in the mother themselves, and then that passes on to the child, or if it is like a predisposition that gets expressed when the child is born.
I think it's very likely both of those pathways to some extent, though I think personally the more impactful pathway is going to be one in which, mums experiences of adversity probably impact her microbiome early in life. I mean, obviously they would, given our data. Those changes may persist into adulthood, they may not, but I think they're definitely going to program things like stress reactivity, vulnerability to depression, which may set up another kind of exposure in the prenatal or postnatal period, which then impacts the child's microbiome. I think that kind of indirect pathway makes more sense with the data that we're getting, but there are certainly a lot of data showing that mum's gastrointestinal microbiome and her vaginal microbiome influence the initial colonies of the child's gut, depending on the mode of birth. So I think they can both play a role, but yeah, I think there's a good chance that there's some other systems that are important to pay attention to there. Yeah. Yeah.
Hi there. Thank you for your talk. That was so exciting. I feel like this is a new field that you're emerging and exploring, so I'm super excited for you.
Thanks.
I have two questions. First is, since the initial results about correlation between the maternal health and the specific bacteria were so specific to certain bacteria, do you think that the first step to using this data would be for like identification of certain mental health outcomes being specifically to maternal like health? Would it be more useful to be like a diagnostic factor, or is it something that people could potentially use as more so like a preventative measure for like maternal health to childhood?
So you're asking if we should use the microbes as a predictor or a treatment, basically. Yeah. So yes, I think you can probably use them as both at some point. I would definitely not say from these data. And the reason I say that is because all of these data are at the genus level. And within the genus, we've got species, and then within the species, we've got strains. And there can be tremendous functional diversity within, within a genus. So there can be some that are pro-inflammatory, some that are anti-inflammatory. So with these type of data, as I mentioned, 16S, there's not a huge degree of specificity that we can get. Second to that, there's a lot of functional redundancy built into the gut. So there are many microbes that have a pro-inflammatory effect. There are many that have an anti-inflammatory impact. There are many butyrate producers, and they live in a community and they like certain environments. So there are lots that love living in the mucus of the gut, and there are lots that love living in the villi. So I think we really need to be very targeted if we're going to use an intervention of the gut microbiome that pays attention to that community structure. And I think that probably a more effective pathway is going to be focusing on communities of microbes that have a particular function in the gut in order to kind of enhance that. So I think it can be used in both ways, but there'll be a lot more data that needs to come about before we could get there.
And second question is, I know, and I'm happy to have learned from your human factors class, which I still think about a lot.
Thanks.
That like the attachment figure is not only always the maternal figure, and so these correlations to the microbiome that you're finding, do you think that there is a specificity between maternal attachment figures and just general attachment figures? Say it would be like a maternal or a community maternal figure.
Yeah.
That's something interesting to look into, or we could kind of say this is mostly for like the childbearing maternal figure.
No, I think there are certain things that are specific to mums. So like vaginal transmission when you're giving birth is obviously specific to mums, and breastfeeding will be specific to, whoever's breastfeeding the child, which is typically the mum. So those things will be specific, but when we live in close proximity to individuals and we engage in behaviors together and feeding and whatnot, those all influence the microbiome. So I think there's a big impact there as well. And then of course, we use the data that we have access to, and often mums are the ones who come into the lab, but we definitely think that the impact of dad on children or whatever other parent on children's stress reactivity and functioning, all of these things that can impact the microbiome will be significant as well. Yeah.
Thank you.
Hi.
Hi, this was an amazing talk. Thank you so much. I was wondering if you or if you're interested in exploring medication use, especially in relation to adversity. So I'm thinking like any antidepressants, anti-anxiety medicine to look at the effects of that on the maternal relationship.
Yeah. So,
do you mind repeating the question? Please?
Oh, yeah, sorry. The question is about medication and whether we control for that and whether that's important to control for. And the answer is yes, it is important to control for. We do collect the data. We do control for it. We don't have sample sizes that are big enough, as in enough kids on SSRIs, for example, to do a sub-analysis and look and see the impacts of that on the microbiome. Though there are labs that do look very deeply at, for example, how does an individual's microbiome composition as they're going on to a drug impact their response to a drug? We plan to do more of that work. We've got a lot of hypotheses about zuranolone, which is the new medication that's approved specifically for peripartum depression. That's an allopregnanolone mimicking medication. There's a lot of background obviously into that, but we've got specific hypotheses about that one drug because that's of interest to us. But otherwise, we're just kind of controlling for medication. Yeah.
So you mentioned the idea that diet and the intervention. One thing that is very different between communities, not microbes or people,
Yes.
is access to food.
Yes.
So for example, we're doing this thing about your cycle A versus other communities. Do you have any data about socioeconomic status and these things?
Yes. So yes and no. So sorry, and the question is basically access to food, access to high quality diets, food deserts, socioeconomic status, how might that impact the microbiome? So we always collect socioeconomic status measures in our lab, even the adversity exposed kids, so kids who are adopted from foster care, they tend to be higher in SES. So we don't really think that there's going to be, well, there's not a lot of variability in SES within our samples. And then the other data, those really big sample sizes that we have, that's from a collaborator in Singapore, also largely high, middle to high SES. So again, not one where we might see a lot of variability in, say, real true nutrition access. There are data in, better samples that are designed for those questions, suggesting that yes, SES does have an impact on the microbiota. And I do think that specifically like food deserts and being malnourished is going to be a really important pathway to look at with adversity exposed youth, but we just don't quite have that sample yet. So I think you need to go to kids who are like actively being maltreated. And obviously that's a really hard sample to research. So all of our data are on kids who are, adopted and thus, fairly well nourished.
Along the same line, what if any would be expected impact of antibiotics to be on this? Because there are kids like, well, I had one that I had so many ear infections at the first couple of years of life, like every month another round of antibiotics. And I was going,
Yeah.
Well, we needed to treat them, but at the same time, we do know that that is a way, to destroy a lot of these.
Yeah, I did want to do a study, though it was just hard to get off the ground, but specifically on kids who are taking prophylactic antibiotics for ear infections, because it is like a healthy sample otherwise that's having a high antibiotic exposure, but it was just hard to to figure that out. So it's funny that you mentioned that. But yes, so antibiotics, absolutely. I mean, that's the point of them. They destroy the microbiome, and that's what we want them to do. And if they stop doing that, we'll all start dying. So they're very important. And they're very important for chronic infections and whatnot. So I definitely want to preface that. But they do obviously have an impact on the microbiome that's, somewhat recoverable. So you are, once you start eating food and, living in your environment, you will become colonized again. But there is clear evidence that the more antibiotic exposures you have, the more opportunity for opportunistic pathogens to enter the gut and antibiotic resistant microbes. So there is an important set of questions to investigate there. We always look at antibiotic exposure in our study. We actually wait for kids to get off antibiotics for a few months before they come into the study. But we don't have great data on like how many antibiotic exposures they've had during their life, mostly because kids in foster care, no one has that data on them. Yeah. Yeah.
While the Zoom audience is thinking about the questions, how to tease out any compensatory or effects between the microbiota itself and whether resilience, so your body's reaction to adversity, affecting the microbiome, and we have granular data to actually tease these things out. Is that an important thing? Or?
So you're asking about resilience to adversity and how that impacts the microbiota?
And then how to tease out when you have these interactions with all these measures, how do you either control or extract these things?
So we definitely have data on resilience, I guess, in the sense of like kids who've been exposed to really traumatic events who don't have elevated mental health problems. We have plans to look at that in relation to the microbiome, but I'm not sure that that's not really your question, is it?
Maybe we can.
Okay. Okay, cool. That sounds good. Okay.
So we don't have any questions.
Okay, great.
So let's thank Bridget again.
Okay, thank you so much.
See you guys.
It might work for you because
Sounds good. All right. See, thanks for coming.